or organs. In aseptic condition, in culture media, in in vitro condition inside the test tube, in vitro condition inside the test tube, a cell or tissue or organ can ability provide a whole plant.
Yes.
Able to provide whole plant. This condition called as this process. This process called as tissue culture, right?
Yes, sir.
Actually, normally how the plants are developed, how the plants are germinated, developed from the single seed, right?
Yes, sir.
Seed. The seed germinated plant. Then how seeds are produced?
After fertilization from the
After fertilization, that means
zygote.
after production of plant, please in mute. Right, thank you.
Sir.
After developed plant, which produces root and shoot and leaf, and finally, when reach maturity stage, produce a flower. Inside the flower, male and female gamete fuse. As a result, after fertilization, zygote embryo seed develop. Again, seed develop. This is time-taking process, right? Isn't
Yes,
it?
sir.
Again, the seeds are produced. In this, some seeds may be viable, some are non-viable. Only viable seeds can produce the plant. Only viable. Not non-viable fail to produce the plant. So we can get only less in number of plants. But in technology, in biotechnology, the only cell or tissue, organ, through the culture media, we can get whole plant. For example, I've taken the 100 cells. I can produce 100 plants only. 100 tissue parts, 100 plants, 100 organs, 100 plants. Then what about time-taking process? Can it take more time or less time? Hello? Hello? Hello?
Sir. Can you please repeat the question, sir?
In tissue culture process, a cell or tissue or organ to produce the plant, whole plant, it take more time or less time?
No, very less time, sir.
Very less time when compared with conventional method.
Yes, sir.
Right?
Yes, sir.
Then what property behind? Inside
Totipotency.
Right, that's it. The totipotency. So what is totipotency?
A cell can able to generate the whole
What I
plant body.
explained so far, this whole process called as totipotency. That is the ability to produce, to plant. A cell have ability to produce the total plant. That ability called as totipotency.
Omnicellular.
Right. If in examination, they may ask, write about totipotency or steps in tissue culture process. You can write what is going to explain now. That is only steps, tissue culture or totipotency process, the same question. In examination, if they ask steps in tissue culture or write about totipotency, question different, but answer same. The answer is, if the answer is the first one, the one by one first, write definition. What is totipotency? What is tissue culture process? After the definition, you have to mention the steps. Steps should be in first one, selection explant. What is explant?
The plant from which we are collecting the tissue.
That's enough. One, explant selection.
Plant should be healthy.
Right. So after selection of explant, the explant should be transported into Inoculated into where the plant grown.
In vitro conditions.
Right, in vitro
Medium.
condition inside the medium. Now you should be select
The proper nutrient medium.
medium. Then okay, explant and select medium.
Inoculation.
Inoculation of
Explant. Callus.
right. Explant. Next, incubation. Next, provide plantlets are transferred
Into the soil or
outer environment or soil, transferred to outer environment. So they are the normal basic steps are transferred to greenhouse. Transferred to greenhouse. Transferred to greenhouse, right? Greenhouse. So they are the totipotency process steps or tissue culture process steps. Don't get confused. In examination, they write about totipotency, write about tissue culture process or tissue culture steps, tissue culture technique, whatever it may be. The different way of asking questions in examination, but answer is only one. Right. First one, explant. First of all, what is explant? Already told you, explant is any part of the plant. Any part during culturing process. If you select any part of the plant, such as leaf is explant, tissue parts explant, pollen grain explant, ovule explant, cells, plant cells explant. Anyone considered as the explant, right? Where is the explant now actually? Explant is the outer environment, right? Out environment, they may get infected with microorganisms. Isn't it?
aseptic conditions. Yes, sir.
Right. So first of all, these explant, maybe shoot tip or root tips, whatever it may be, should be sterilized.
Yes.
Should be sterilized with detergents or some of the chemicals. The wash in chemicals or detergents, the
Spirit.
explant should be sterilized.
Alcohol.
Alcohol also should be sterilized. So after sterilized,
Sanitizer.
sterilized means that's it. That's the same meaning. Right. First of all, selected explant should be sterilized with the help of chemicals or alcohol. Selection completed. First of all, sterilization also completed. After that, select proper medium. Proper medium. The most of the scientists utilize the Mauritius Cook medium. Mauritius Cook medium, MS medium. MS medium for supporting agar gel for growth hormones, nutritions, some nutritions also. Some nutritions also. They are included in MS medium. Due to the high nutrition value, there is a chance to infected with microorganisms, right?
Yes.
So ensure these also not to infected with microorganisms. So they should be sterilized. So now the explant sterilized and selected MS. Right. Right, where we are? Right, okay. Extra sterilized and selected medium MS group, MS media, that is with help of nutrients and also growth hormones, oxygen, cytokines, they are all provided inside the medium. Now both are ready. Now third step, inoculate explant. Inoculate means entry. Entry transfer the explant into the medium. That is called inoculation. The inoculation should be in the specialized sterilized room, laminar airflow. In laminar airflow, chamber, inoculation process carried out. Of course, as the plant explant outside, there is a chance to get in microorganisms.
Contaminated.
Medium also, due to the having the much rich nutrients, it is easily infected microorganisms. So these both are sterilized. The first one sterilized chemical, the second one sterilized in autoclave instrument. So anyway, after both are sterilized, right? Both are sterilized, the explant introduced into the medium. While you introduced into the medium, there is a chance to infect with microorganisms. If this process carried out outside, not well maintained, aseptic condition, unaseptic condition, if you do outside the weather, while the inoculation, while the explant introduced into the medium also may get infected. That's why this process should be in the aseptic condition, aseptic chamber. This aseptic chamber should be laminar airflow. In laminar airflow, inoculation process. Due to inoculation, any one explant, whether it is tip or fruit, cell, tissue, anyone introduced into the medium. What happened after introduced into the medium? Already inside the medium, the enough nutrients are there, enough phytohormones are there.
Cell division occurs.
Now, from this explant, growth started. The first undifferentiated parts are developed. That group of cells, just group of cells are developed. This group of cells is called
callus.
Callus, not thallus, callus.
I said callus only, sir.
Okay, right. Callus. So to develop the callus, now add extra cytokinins and oxygens phytohormones. Then only from the callus, morphogenesis takes place. Morphogenesis means, what is genesis?
Production.
Production. Morpho means?
Morphological characters like root, stem, leaf.
Not morphological character, morphological structure produced. What are the morphological structure?
Root, shoot, leaf, root, stem, leaf.
Right, root, stem, leaf. The mainly first due to the activity of cytokinins, the oxygens ratio, the same way, but that means the ratio difference. Rich cytokinin, less oxygen, roots are erased.
Yes.
This process called as rhizogenesis. In inoculation process, rhizogenesis. Rhizogenesis, root formation. At the same way, oxygens, high, low cytokinin, collagenesis also take place. That means stem and leaf. Stem and leaf. That is collagenesis. Anyway, the morphological parts are developed. Morphological parts are developed. After formation, morphological parts, small, simple plantlet, the 1 or 2 weeks, it is undergoes to incubation period. After incubation period, after 2 weeks or 3 weeks, now the plants are transferred to the plant, that is transferred to the pot, red clay pot. After that, transfer to the greenhouse. The finally planted in field. This is the tissue culture at process. Mainly 5 steps. First, select excellent and sterilize them. Second, selected medium, sterilize. And third, inoculation in sterilized room. Sterilized room. After utilized nutrient and phytoorganiments produces developed morphological structure, root, shoot, leaf. After developed small plantlets, after incubated several weeks, 1 to 3, 2, 3 weeks. Now it is transferred to the pot, soil clay plot. Now finally transferred to the outer environment. Now we get a whole plant. This is the property of totipotency and process of tissue culture. Is it clear?
Yes, sir.
Right. Right, in this process, the application of totipotency or application of tissue culture. Let's see the application totipotency or tissue culture. First one, production of synthetic seed. Production of synthetic seed. How the seeds are produced? Already asked you. How the synthetic seed, how the seeds are produced? In general, seed production.
Way.
In natural way.
Fertilization, sir.
Right. Fertilization, male, female gamete, zygote, embryo, seed. That's it. What is synthetic?
Without fertilization. Sorry, may not be. Without.
That's right. There in synthetic seed production without fertilization. In vitro condition, during process of tissue culture, in a media, embryos are formed. Such embryos called as somatic embryo. Somatic embryo formed. Like a call as somatic embryos are formed.
Okay.
After formation, these somatic embryos, this somatic embryo covered with chemical. This is somatic embryo, and this somatic embryo covered with chemicals or encapsulation with some chemicals. This calcium arginate, sodium arginate, or calcium. Sodium arginate. And also
No.
So during tissue culture process, the somatic embryos are formed. Somatic embryo. Because why they are somatic embryo? They are not a, these embryos formed by not by fertilization. These are formed by only tissue culture process. That's why it is called somatic embryos. And these somatic embryos covered with some chemical layer, encapsulated with layer, calcium, sodium, oxygenate. Hello. 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 Hello.
So,
Is it you?
your voice is breaking, sir.